There is a place on this planet where the pressure is so extreme that calcium carbonate, the stuffed bones and seashells are made of, starts to dissolve. Below 6,000 meters in the ocean, rigid skeletons become a death sentence. So how is a fish living at 8,000? The hadal snailfish solved the problem by doing something no other vertebrate has done. It carries a mutation that stops its own bones from fully hardening. Its skull has gaps that never seal shut, and instead of collapsing under the weight of the ocean, its soft, flexible body absorbs it. If you'd asked any marine biologist in the early 2000s about vertebrate life in the hadal-zone, they would have told you nothing with a backbone belongs down there. The pressure alone should crush internal organs. The darkness is total. The water sits just above freezing, and the seafloor at those depths is a barren stretch of sediment, miles from sunlight and photosynthesis. They had no idea what was waiting. The hadal-zone starts at 6,000 meters and stretches down to nearly 11,000. Only a handful of trenches on Earth reach that deep, and the Mariana Trench in the western Pacific is the deepest of all. Its lowest point, the Challenger Deep, sits roughly 11 kilometers below the surface. The pressure down there is over 1,000 times what you feel standing at sea level. A styrofoam cup, lowered to the bottom, comes back the size of a thimble. Metal groans and cameras implode. Yet life persists. The hadal snailfish, known to researchers as Pseudoliparis swirei, was first formally described in 2018 after being collected from the Mariana Trench at depths between 6,000 and roughly 8,000 meters, according to the Nature Ecology and Evolution study that published its genome. This fish is small, pale, and translucent, almost ghostly. You can see its organs through its skin. It has no scales. Its body is gelatinous, closer to jelly than muscle. It looks like something that should fall apart in your hands. It is the exact opposite. Its secret sits inside its DNA. The gene responsible for producing osteocalcin, a key bone protein, carries a frameshift mutation. According to that same genome study, this mutation knocks the gene out of service before it can finish its job. Osteocalcin normally helps bones absorb calcium and harden into rigid structures. Without a functioning copy, the snailfish's skeleton stays partially cartilaginous. Its bones remain soft. Its skull never fully fuses. And 3 other genes involved in bone mineralization, including TMEM-thiopharma1 and TMEM-TAMDONGBA, have also been lost entirely from its genome. This fish did not just weaken its skeleton. Over evolutionary time, it deleted the instructions for building a hard one. That matters because a rigid skeleton at 8,000 meters would be a liability. Calcium carbonate, the mineral that makes bones and shells hard, becomes increasingly soluble as pressure rises and temperatures drop. Past the carbonate compensation depth, roughly 4,000 to 5,000 meters in the Pacific, calcium carbonate dissolves faster than it can accumulate. Every shell, every bone fragment, every piece of coral that sinks past that line eventually disappears. Sediment samples from the hadal-zone contain almost no calcium carbonate at all. The ocean floor down there is soft clay and siliceous ooze. The snailfish bypassed the problem entirely. It evolved to stop building the thing the ocean was trying to destroy. Pressure does not just attack bones. It warps proteins. At hadal depths, the water column pushes molecules so close together that proteins begin to unfold and lose their 3-dimensional shape. A misfolded protein cannot do its job. Enzymes stop catalyzing reactions. Cell membranes they destabilize. Ion channels misfire. For most fish, the biochemical limit sits somewhere around 5,000 meters. Past that, their cellular machinery simply stops working. The snailfish beat this with a molecule called TMAO, trimethylamine N-oxide. TMAO is a piezolyte, a pressure-resisting chemical that stabilizes protein structure under extreme compression. It works by strengthening hydrogen bonds between water molecules surrounding each protein, forming a kind of molecular scaffolding that holds everything in place even as pressure tries to squeeze it apart. According to a study published in the Proceedings of the National Academy of Sciences, TMAO concentrations in deep-sea fish increase linearly with depth. The deeper the fish lives, the more TMAO it packs into its muscles and organs. The hadal snailfish, living near the theoretical maximum depth for any fish, carries some of the highest TMAO concentrations ever measured in a vertebrate, and it can produce so much of it because of another genetic trick. According to genome research published in PLOS Genetics, the hadal snailfish has 5 copies of the FMO3 gene, where the average fish carries 1. The FMO3 gene encodes the enzyme flavin-containing monooxynase 3, which converts trimethylamine into TMAO. 5 copies means the snailfish can crank out this protective molecule at a rate no other vertebrate can match. On top of that, the snailfish's gut is loaded with bacteria that generate the raw material, trimethylamine, feeding the whole system from the inside. The fish does not just tolerate pressure, it runs an internal chemical factory designed to resist it. So you have a fish with a skeleton that refuses to harden and a protein shield that multiplies under pressure. Those 2 adaptations alone would be enough to earn it a place in any biology textbook. But the snailfish is not the only organism that has rewritten its own rules to survive in the hadal-zone. The deeper you go, the stranger things get. If you're finding this interesting, consider subscribing. There are more deep dives like this one on the channel every week. Scattered across the floor of the Mariana Trench, you will find xenophyophores. They are single-celled organisms, 1 cell, and some of them grow to the size of your fist, around 10 centimeters across, sometimes even larger. They have been documented at depths exceeding 10,000 meters, making them among the deepest living organisms ever found. They belong to the foraminifera, a group of amoeba-like protists, but they look nothing like any amoeba you have seen under a microscope. They build elaborate outer shells called tests by gluing together tiny fragments of sediment, mineral grains, sponge spicules, the skeletons of other dead organisms. The result looks like a lump of seafloor debris, but inside is one continuous cell containing thousands of nuclei. They are the largest single-celled organisms known to science. They thrive in a place where multicellular animals struggle to survive. Xenophyophores are not just living down there. They are shaping the habitat around them. Their tests create small ridges and pockets on the otherwise featureless seafloor, providing shelter for tiny invertebrates and trapping organic particles that other organisms feed on. Where you find xenophyophores, you find more biodiversity around them. They are the unintentional architects of the abyss. Then, there are the amphipods. Hirondelia gigas is a tiny crustacean, roughly the length of a paperclip, and it dominates the scavenging community in the deepest trenches on Earth. Drop a baited trap to the bottom of the Mariana Trench, and within hours, these amphipods will swarm it by the thousands. They are aggressive, efficient, and everywhere. In a zone with almost no visible life, they are the closest thing to a crowd. But food at 11,000 meters is desperately scarce. Whale carcasses and dead fish sink down occasionally, but those events are rare and unpredictable. That is not enough to sustain an entire population year-round. According to a study published in PLOS One, researchers discovered that Hirondelia gigas has evolved its own cellulase enzyme, a molecule that can break down cellulose, the tough structural material in wood and plant matter. When chunks of wood sink to the trench floor from distant coastlines, river systems, or storms, these amphipods can actually digest them. No other deep-sea amphipod had been documented with this ability before. The enzyme, which the researchers classified as a family 31 glycosyl hydrolase, is unlike any cellulase found in related species. It performs even better under the extreme pressure and near-freezing temperatures of the Challenger Deep than it does at surface conditions. Evolution did not just help these amphipods tolerate the trench, it optimized their biochemistry for it. This is what makes the hadal-zone so disorienting to study. The rules that apply everywhere else in the ocean break down here. Bones become a disadvantage. Proteins need chemical bodyguards to hold their shape. Single cells grow to the size of a golf ball. Shrimp-like creatures evolve the ability to eat driftwood. Every organism at these depths has rewritten some fundamental part of its own biology to exist in a place that seems engineered to prevent life. The snailfish rewrote its skeleton. The xenophyophores rewrote what it means to be one cell. The amphipods rewrote their entire diet at the molecular level, and all of them did it in total darkness. under pressures that would crush a submarine hull on a seafloor that most humans will never see with their own eyes. We have better maps of the surface of Mars than we do of the hadal trenches. Most of the ocean floor remains unmapped and unobserved. The creatures we have found so far, the snailfish with its soft skull, the fish-sized single cells, the wood-eating amphipods, were discovered only because someone lowered a camera or a baited trap on a cable and waited. The hadal-zone is one of the smallest biomes on the planet. It may hold some of the most unusual biology anywhere on it. And we have barely started looking. The Mariana Trench is not empty. It never was. It just took us decades of engineering to build something that could survive long enough to see what was already living there. If you want more stories like this, there are plenty on the channel.